I'm Karen Wishner, professor here at the Graduate School of Oceanography at URI. I've been here since 1980. I'm a zooplankton ecologist and biological oceanographer. I've been studying zooplankton throughout the water column. Zooplankton are the small animals in the water column that are the food for the larger fish. Right now, what we're working on are regions of the ocean called oxygen minimum zones. These are areas which have naturally very low oxygen concentration. In this case, these low oxygen regions of the ocean are naturally present. They've been present through geological time, that is millions of years. And so the animals living in them and at their edges are highly adapted to those conditions. This is the upper layer where there's a lot of oxygen. You have a lot of animals both day and night here. There's a day column and a night column. And then in this very low oxygen area, we call it the OMZ core, the oxygen minimum zone core, you have very sparse populations. But what you see in the daytime are these little fish. These little gray things are small fish that have moved all the way down here several hundred meters. This net represents the depth between 350 and 550 meters, so they've moved all the way down there in the day. So certain animals are capable of living part of the time in this low oxygen region, and then these animals will move up at night. At this lower boundary, you have a whole suite of unique organisms that are just positioned in that boundary. So the animals are really keying into a very specific oxygen level. As you look in finer detail, you see that your species are different, but still reacting to the same gradients. And often people will use very large depth intervals and essentially mush all of this together because they've sampled through a large range. When you start to sample smaller depth intervals, then you start to see all these layers and these gradients, and you learn what parts of the environment they're responding to.